¿Listo?
Bueno,
¿Habilitado?
comenzamos nomás, entonces. Bueno, muchas gracias por asistir a este segundo webinar de la Sociedad Argentina de Embriología Clínica. Hoy nos acompaña la doctora Mina Popovic. Ella es doctora en Medicina y Ciencias de la Salud por la Universidad de Gante en Bélgica. Antes de sus estudios de doctorado, trabajó como embrióloga clínica durante más de 5 años en Guinea, Australia, miembro del Grupo de Interés Especial de Células Madre de la Sociedad Europea de Reproducción Humana y Embriología. Mina tiene amplia experiencia en el área de embriología y genética reproductiva, con más de 20 publicaciones científicas, y ha dedicado sus últimos años al estudio de mosaicismo cromosómico en embriones humanos. Actualmente es miembro del staff de investigadores en la clínica Eugene de Barcelona, España. Mina, I would like to thank you again. Thank you very much for accepting our invitation and joining us today in this webinar. As I mentioned before, we will ask the question at the end of this presentation. And if you're ready, let's start it.
Thank you very much. Thank you very much for the kind introduction. And I would like to sincerely thank the Argentinian Society of Clinical Embryology for this invitation. And good afternoon to everyone, good evening, good morning. I'm not sure where everyone is at the moment that they're tuning in, but I would like to extend a warm welcome to everyone joining the webinar. I do apologize that I'm not speaking in Spanish, but it is quite difficult for me, even though I am living in Barcelona, but it's only been for a short time. So hopefully next time, but you'll have to bear with me. But my topic today on mosaic embryo transfer in Pgta is, of course, quite a controversial one, and I'm sure you're aware of this. And it has recently led to a lot of heated debates in the field of pre-implantation genetic testing. And I'm hoping that after this presentation, you'll really be able to also see why for yourself. So maybe also after this presentation, you'll be somewhat disappointed with what I have to say, because my answer regarding the diagnosis of mosaicism and the transfer of mosaic embryos is unfortunately not entirely straightforward. However, I do hope that by discussing and debating and educating about mosaicism, we can really come a little bit closer to moving away from this uncomfortable uncertainty we are currently facing, both as embryologists, as clinicians, as genetic counselors, regarding mosaic embryo transfer in Pgta. So as I've been researching this phenomenon of mosaicism in human embryos for a few years, this has actually made me realize that we must approach the issue from several perspectives. We must, of course, understand the biology, but we also must remain extremely mindful of the technical limitations of Pgta. And finally, of course, we have to put everything into a perspective from a clinical standpoint. And this is the way I will also be discussing today. So we're all familiar with the fact that human development is remarkably inefficient, and this inefficiency is largely attributed to embryo aneuploidy, and it is reflected in the high incidence of preclinical losses during the first week following conception. So ultimately, fewer than 30% of all fertilization events will actually result in a successful pregnancy. And this is even the case for young fertile couples. So while the risk of embryo aneuploidy actually remains quite high across all maternal ages, it is of course well known that it becomes even greater with advancing maternal age. However, what has been suggested is that pregnancy rates remain stable across all age groups if a euploid embryo is transferred. And this has led to this notion that with the selection of euploid embryos, theoretically, this should lead to a reduction in the risk of implantation failure, miscarriage, and aneuploid conceptions. And accordingly, this should also reduce the time to pregnancy. And ultimately, if we select embryos free of abnormalities, this should improve clinical outcomes. And this is really the premise for Pgta. Of course, we know Pgta is globally used quite a bit at the moment, varies across the different areas of the world, but the latest technology and the most accepted technology for Pgta currently is next generation sequencing combined with trifecta and biopsy. And I'm sure you're all very aware of this. However, what complicates Pgta from a biological perspective is that the origin of chromosomal errors in human embryos is varied. So they may be derived during the process of gametogenesis, which will result in an abnormal egg or sperm, or they may be derived in the mitotic divisions that underlie embryogenesis. And meiotically derived abnormalities will generally affect all embryonic cells uniformly. But on the other hand, these post-zygotic errors actually lead to mosaicism. And this is the presence of chromosomally distinct cells within a single embryo. Mosaic embryos may harbor both euploid and aneuploid cells, so we can see a mix of both normal and abnormal cells, or they can be composed entirely of abnormal cell populations. Of course, clinically, what we are most interested in, or is the most relevant, are these euploid-aneuploid mosaics. And when we actually talk about mosaicism in a clinical setting, we really are referring to this type of mosaicism. Again, from a biological perspective, what has been assumed is that mosaicism is less pervasive as development progresses. So we frequently do describe mosaicism during pre-implantation development. However, we see these rates dropping throughout development. And mosaicism is really only diagnosed in less than 2% of prenatal samples. And true fetal mosaicism is identified in only around 0.4% of cases and 0.2% of live births. So then why all this controversy surrounding mosaicism? Well, while we did encounter mosaicism at the blastocyst stage of development much less frequently using previous technologies for Pgta, the implementation of next generation sequencing has really led to an increase in reports of mosaicism. And what I mean by mosaic profiles. So here, these are just some profiles following next generation sequencing. So this is what we're typically looking at. So on the bottom, we can see the chromosome number, and here the autosomes have been presented. And on this axis, we see this log2 relative copy number. Now, sometimes this is presented as just copy number. So we see number 2, 3, and -1. In this case, we're using a log scale, but it's essentially representing the same thing. So the reads here that are mapping around 0 indicate euploidy in all of these positions. However, if we have reads at -1, this indicates a monosomy, and at around 0.5, this is indicative of a trisomy. So in the case of mosaicism, we see a profile like this, where we see a suggestion of an abnormality. However, it's not quite reaching the threshold of what we would consider a uniform aberration, so something that is present in all of the sampled cells. And mosaicism is diagnosed for whole chromosome abnormalities, as we see here, but it's also diagnosed for segmental abnormalities, so where only part of the chromosome is actually affected. So this higher frequency of mosaicism that has been detected by next generation sequencing has really raised a lot of uncertainty surrounding the accuracy of a trifecta biopsy. If we do assume that mosaicism is much more prevalent at the blastocyst stage than we thought, then we could be potentially incorrectly classifying embryos, because of course we assume that Pgta relies on the chromosomal constitution of the biopsied cells being representative of the embryo. So if mosaicism was present to this extent, then this could undermine the principal strategy of improved embryo selection. Now, what we also have to be very much aware of and very realistic about is the technical limitations of Pgta. And what I'm specifically referring to is the limitations of next generation sequencing performed on a low number of cells. So really, this low DNA input remains a major challenge for pre-implantation genetic testing. And what I mean by this is I'll give you some background. So basically, when we take cells from the embryo, we have to extract DNA from these cells. However, this DNA is actually not enough to be sequenced. So the sample actually needs to undergo a whole genome amplification step. And in this step, we generate many, many DNA strands. Then these strands are actually fragmented, and we are literally left with millions of sequences. And these fragmented sequences, also known as sequencing reads, these are actually sequenced. And although to us this is just a jumble of millions of sequences, of course, with bioinformatic algorithms, we can align these to a known sequence of the human genome. And we can tell where each of these reads actually belongs along the genome. So what we hope for, of course, is an even amplification where all of these reads have nicely, evenly amplified. And then we can clearly see if we have an additional number of reads, a trisomy, or if we have less reads, this would be indicative of a monosomy. However, this process of whole genome amplification is actually not perfect. And what often happens is that we have biased amplification, where certain parts of the genome are actually over or underrepresented. And what this actually leads to is technical artifacts. And these technical artifacts of amplification or the sequencing may in fact be indistinguishable from mosaic results. So we have to be really mindful of this. And for this reason, platform validation is really critical. But even with platform validation at the back of our minds, we must always remain aware of the limitations of next generation sequencing on these low cell numbers. So for this reason, really blastocysts that are diagnosed with mosaicism based on a single ultrafactored biopsy can only really be considered at risk of being mosaic. So in addition to the technical and biological factors that play a role here, there are also a lot of inconsistencies in the diagnosis and the interpretation of mosaic results across different centers. And this is very nicely demonstrated here in these two studies. So this was a recent study by Manuel Viotti, which I will actually mention a bit later in my talk as well, which actually nicely shows this proportion of Pgta categories that are diagnosed across different centers. And what we really see is varying rates of euploidy and also varying rates of mosaicism diagnosed across the centers. And in this study by Santiago Munet and colleagues, they actually show that euploidy rates really vary among centers as well. And they show that this is largely due to differences in reporting mosaicism. So in the centers that do report mosaicism, we actually see a trend towards lower euploidy rates. Whereas centers that actually did not report mosaicism or just classified embryos as abnormal or euploid, we see higher euploidy rates and actually even a trend for higher ongoing implantation rates. However, what this really demonstrates is that a standardized approach in diagnosing and reporting mosaicism is really lacking. And this is very important when we are looking at data that is published following the transfer of mosaic embryos and any data actually on mosaic diagnosis. So, okay, let's assume that an accurate mosaic diagnosis can be achieved. Well, then still this question regarding the implantation and the developmental potential of mosaic embryos still remains. And even though we have been faced with this concept of mosaicism for a few years now, there is still no clinical consensus on whether an embryo that's diagnosed as mosaic should be transferred. So we decided to tackle this from a more fundamental perspective, and we actually used this extended in vitro culture system to culture mosaic embryos. So what this system actually allows us to do is keep the embryos in the lab up to 14 days. And we optimized this system in our lab, and we were able to see phenotypes that are associated with post-implantation development. So this system actually, in this system, we plate embryos on day 6, they form outgrowths, and then we can grow them, and we grew them up to day 12. And using this system, we actually cultured embryos of known chromosomal profiles. So we cultured abnormal, mosaic, and euploid embryos. And as I said, we grew these embryos to day 12, and then we actually collected these embryos again for sequencing and for sequencing. And in fact, for a limited number of embryos, we were actually able to separate them into inner cell mass and trophectoderm-derived portions at day 12. Unfortunately, we couldn't do this for all the embryos because this distinction between these two parts of the embryo is not always very obvious. So we only did it in the cases where we were quite confident of these two structures. So what we first did is we looked at the developmental outcomes. So what we observed is that around 50% of the embryos actually remained attached and viable throughout this extended culture, whereas the other 50% actually showed signs of cell death and degeneration, and they detached by day 12. What we then did is we compared these developmental outcomes to the blastocyst chromosomal profiles. And what we observed is that embryos that were euploid, those with trisomies, duplications, and mosaic embryos, were significantly more likely to remain attached throughout the extended culture, whereas embryos that were diagnosed with monosomies, deletions, or multiple abnormalities were significantly more likely to detach. Now, after this, we then also compared the blastocyst chromosomal profiles to the chromosomal profiles on day 12. And what we observed here, actually, that was quite reassuring, is 100% concordance for uniform numerical aneuploidies. So what I mean by numerical aneuploidies are whole chromosome abnormalities. And as we see here, this embryo was diagnosed originally with the trisomy 16. This is apparent that it is present in all of the cells, so it is uniform. And we actually see this abnormality persisting throughout development. However, when we looked at uniform segmental aberrations, so those affecting part of the chromosome, we actually could not confirm them in all cases. And as we see here, we have 2 embryos with duplications, and we see no evidence of these duplications at day 12. And in fact, in this case, we had an embryo which we were able to segregate into 3 parts at day 12. What we also observed is that around 58% of the embryos that were diagnosed as mosaic generated viable outgrowths. And in fact, around 70% of these embryos that were diagnosed as mosaic also generated euploid outgrowths. So as we see here, this is an embryo diagnosed with a mosaic segmental abnormality, so a segmental deletion. And we see no evidence of this aberration at day 12. Euploid outgrowths were observed for both numerical, so whole chromosome, and segmental mosaic diagnosis. So in this example here, I'm just showing an example where we actually see both in the same embryo. So as you can see, we see no evidence of these abnormalities at day 12. In some cases, we did confirm the mosaicism though. As we see in this example here, we had an embryo with a mosaic trisomy 3 that was diagnosed at the blastocyst stage. And in fact, at day 12, this abnormality seems to be present in all of the cells. And in this example here, this is actually a very interesting one because here the embryo was diagnosed with a mosaic trisomy 4 at the blastocyst stage. However, at day 12, we see a reciprocal monosomy 4 in the cells. And what this actually is really indicative of, and it serves as biological evidence of a mosaicism. This is because we can really see this evidence of a mitotic era where we see 2 cell populations. Some of them have the gain of the chromosome, whereas in others we see a loss. So overall, what we showed was that euploid blastocysts and those with chromosomal gains were more likely to remain viable. Also, monosomies were significantly more likely to detach. And what was reassuring was that we observed 100% concordance for uniform aneuploidies and euploid diagnosis. However, uniform structural abnormalities, segmental abnormalities, were not always confirmed. And a high proportion of blastocysts diagnosed as mosaic generated euploid outgrowths. So we were quite pleased with our results because they really were reflecting and echoing prenatal data. So the fact that we don't see many monosomies at the prenatal stage is also corresponding to our results. But we were also happy to see this concordance for uniform aneuploidies. And what this actually suggests is that Pgta is quite good at diagnosing uniform whole chromosome aberrations. And these are actually mostly indicative of clinical outcomes. And this was nicely demonstrated in this prospective non-selection study, which was recently published by Teagues and colleagues. And what they actually did was that they tried to predict the accuracy of a trabectedin biopsy, the accuracy of the biopsy itself to predict the success or the failure of a healthy live birth, so this clinical accuracy of the biopsy. And what they actually did is that they transferred embryos which they had previously tested. However, they did not reveal the results until they knew the clinical outcomes. And what they actually showed that euploid embryos, 65% of euploid embryos generated live births, whereas 0% of aneuploid embryos led to live births. So this really shows that no viable pregnancies were obtained following the transfer of blastocysts diagnosed with uniform whole chromosome abnormalities. And we can see this nice summary of the results here, but what is further interesting is that they also reported on a very small number of embryos that were diagnosed with whole chromosome mosaicism. And what they actually see is very similar results to the euploid embryos. Now, of course, it's on a very small number of embryos, but still worth a mention. So over the past years, really this knowledge regarding the developmental capacity of blastocysts diagnosed as mosaic has predominantly stemmed from clinical outcome data following Pgta. And as we can see up until now, there have really been several studies that have looked at outcomes of embryos that were categorized as mosaic, and they matched these outcomes to euploid counterparts. And while this data is still relatively limited, what current findings are consistently suggesting is that blastocysts that are classified as mosaic do have the capacity to result in viable pregnancies. However, they also do entail higher rates of miscarriage. And this was also confirmed in the study by Viotti and colleagues, and this is actually outcome data from the largest study following the transfer of 1,000 mosaic embryos. And they actually also see that mosaic embryos implanted less and they miscarried more, but they did have the capacity to result in viable pregnancies. So with this data, we still had this uncomfortable uncertainty on what to do, and really, should we transfer embryos that are diagnosed as mosaic? And as a result of this uncertainty, there have been various approaches. So the first one has been the conservative approach. So in this approach, clinics are actually not reporting mosaicism, or actually they are classifying any samples with more than 30% abnormal cells as clinically unsuitable. And this threshold varies between laboratories, and some labs are actually classifying all mosaic diagnosis as clinically unsuitable. And this approach is really based on concerns of the unknown potential risks of classifying embryos with some aneuploid cells. And also what this strategy makes perhaps easier or more straightforward is the patient counseling, because it is always more straightforward to counsel patients about the absence of a finding rather than the presence of a finding. But what are the limitations of this conservative approach? Well, we have an increase in the risk of having no blastocysts available for transfer. And this is particularly limiting for patients with only a few embryos available. And ultimately, this may reduce the probability of pregnancy for the patient. This was in fact nicely demonstrated by this study, which actually compared ongoing pregnancy rates following next generation sequencing-based Pgta and RACGH-based Pgta. And what they actually showed is higher ongoing pregnancy rates following next generation sequencing Pgta. However, what they also showed is that patients that underwent Pgta with NGS had significantly fewer embryos available for transfer. And this was due to this exclusion of embryos that were diagnosed as mosaic. So because of these legitimate concerns of actually discarding potentially viable embryos, this new emerging approach we have come to this new approach. And basically what it entails is that mosaic embryos that are classified as mosaic should form a third category of embryos in our diagnosis, and these are embryos with reduced potential. So this notion is based on the fact that embryos, mosaic embryos, are inevitably being transferred during routine ART cycles. And we also see an exceptionally low rate of pregnancies and live births that are affected by mosaicism. So really an unknown risk does not necessarily equal a high risk. However, there are also limitations of this new emerging approach. And this is, in fact, that this concept of mosaicism is extremely difficult to grasp for us, let alone the patients. So only around 30% of patients would actually accept the transfer of an embryo diagnosed with mosaicism. And also what we are seeing, and this was from a recent study, was that fewer than 3% of embryos that are actually diagnosed as mosaic are selected for clinical use. On top of that, diagnosing embryos as mosaic adds another layer of complexity, and that is which characteristics of a mosaic diagnosis actually correlate with clinical outcome. Is it the proportion of aneuploid cells in the biopsy? Should we consider the nature of the aneuploid involved, the type of mosaicism? And should we consider the identity of the aneuploid chromosomes present? So I will try to tackle some of the evidence that exists for each of these different characteristics. But I do want to warn you that the evidence that we currently have is really based on studies with retrospective design. And these are subject, of course, to patient and embryo selection bias. We know that embryos that are diagnosed with mosaicism are only really considered for transfer if no euploid embryos are available. So this could lead us to a selection bias towards poorer prognosis patients. And we really need more prospective non-selection studies to really be able to have a good grasp of the relevance of these characteristics. Nevertheless, what is known so far? So regarding the proportion of aneuploid cells in the biopsy, so this degree of mosaicism, actually, the study that was recently published on the 1,000 mosaic transfers, which I've mentioned now a few times, showed that embryos that were diagnosed with high levels of mosaicism actually had poorer outcomes, as you can see here. However, what was very interesting was that this did not apply to embryos diagnosed with segmental mosaicism. So it was only really seen in the case of whole chromosome mosaics. So with segmental mosaicism, we see no difference compared to euploid embryos in clinical outcomes. And this was somewhat confirmed in this study that was recently published by Capalbo and colleagues. Now, these are preliminary results, so this study is yet to undergo peer review, but the results are actually quite interesting. And this group showed that embryos with low mosaicism and moderate mosaicism, so anything with less than 50% abnormal cells, actually had equivalent results to transferring euploid embryos. Now, as I said, this study still hasn't been published, but it would be very interesting to know what types of mosaicism were actually diagnosed here. So now we get to the nature of the aneuploid involved. So this type of mosaicism, and I already touched on this a little bit. So what has been suggested is actually that segmental mosaics have the most favorable outcomes. And this is regardless of whether they are present in a low or a high degree. Next, we see that the number of chromosomes affected did correlate with poorer outcomes. So embryos that are classified as complex mosaics had worse outcomes than those with just one chromosome affected. However, what we do see is regardless that this level of mosaicism seems to have a greater effect. So in all cases, low-level mosaics had more favorable outcomes compared to those diagnosed with high-level mosaicism. And finally, we get to the identity of the aneuploid chromosomes present. So the original guidelines that were developed by the Pre-Implantation Genetic Diagnosis International Society suggested that embryos that are classified with high risk or mosaicism affecting high risk chromosomes should actually have the lowest priority. And since these guidelines have been further amended to again suggest that low-level mosaics should have a higher priority compared to high-level mosaics. And in the study by Biopsy and colleagues, what they actually show is that viable pregnancies have been achieved from mosaic diagnosis involving all of the chromosomes. So here we do not see that any chromosomes are worse off. But again, when we turn to prenatal samples, there is some evidence. So these are studies based on the analysis of prenatal samples, specifically this study by Francesca Gratti and colleagues, and they analyzed really a large number of CVS and products of conception. And what they actually showed is that mosaic trisomies of high risk, so those affecting chromosomes 13, 14, 16, 21, and X, should really be avoided. And then in a further study that was based on the analysis of amniocentesis samples, this group actually showed that mosaic monosomy X and mosaic trisomy 21 are actually the most common. So perhaps these should be, this reiterated that these should be avoided. But what this study showed, which was quite interesting again, is that really true fetal mosaicism is present in really a low level, in a low ratio. What we do have to remember, though, is that these chorionic villi sampling, amniocentesis samples, placenta, and newborn mosaicism may actually not reflect the Pgta analysis, where we have a few cells sampled. So we really have to continue looking at the mosaicism at the blastocyst stage. And what these studies do reveal is that IVF conceptions are not really at a greater risk of true fetal mosaicism and segmental abnormalities compared to natural conceived pregnancies. So this is quite reassuring. Nevertheless, we should be cautious, and I would like to just mention this study that was recently published that actually showed the birth of a baby with mosaicism that resulted from a known mosaic transfer. So here we had a blastocyst that was diagnosed with mosaic monosomy 2, and this embryo was transferred. It resulted in a pregnancy. However, the amniocentesis revealed a mosaic trisomic karyotype for chromosome 2. So we actually again see this reciprocal abnormality, and the newborn blood actually showed again a mosaic monosomy 2. So this is really evidence that my biological mosaicism does exist and that we should be careful. So just to summarize, and these are a few considerations regarding the diagnosis of mosaicism and the transfer of mosaic embryos. From a laboratory perspective, we have to remain aware that suboptimal blastocyst biopsies may create false mosaic results. And this is because suboptimal biopsies can lead to DNA degeneration, and this can this noise, in fact, and these artifacts of amplification may be indistinguishable from mosaic results. On top of that, we have to realize that there is a subjectivity in diagnosing mosaicism. We see this with all these varied results that we're observing across the different clinics. And really the analysis software that we had previously for Pgta is really designed to classify samples as uniformly euploid or aneuploid. So we should be really aware of this. So standardization of clinical and laboratory practices for achieving consistencies is really crucial. And what we should also consistently revise and reconsider are these recommendations for transferring mosaic embryos. So as more and more data becomes available, we will become more knowledgeable, hopefully, and this will really allow us to make more informed decisions. Of course, patient counseling is crucial of patients undergoing, of course, a mosaic embryo transfer, but also just of Pgta patients. And they really should be aware that potentially viable embryos may be discarded, even if under the premise of mosaicism, or even if mosaicism is not diagnosed. So overall, we have to continue to evaluate biological variants to define more specific criteria. We need to find ways to dissociate this biological variance from the technical variability that we are limited by. And also we have to continue to perform large studies, both retrospectively and prospectively, and hopefully this will give us more insights. So I would like to thank you all very much. And I would just like to acknowledge my collaborators from UZ Ghent from Belgium, and the Ghent Fertility and Stem Cell team, the Center for Medical Genetics in Ghent, Belgium, and also the Leiden University Medical Center in the Netherlands. And thank you all very much for your attention. And I'm really looking forward to the discussion.
Thank you, Mina, for your nice presentation. I think that all of we are very excited about your data. And we're going to start with the discussion, and we have some questions for you. Here, Constanza is asking you if we have already transferred mosaic embryos, and if you have a live birth, how are you sure that this baby is going to be safe? Do you make any studies in the live birth after mosaic embryo transfer?
Yeah, so this is a very good question, and it is quite challenging, and not many studies have addressed this, because of course we know that in clinics we are not always following up all of the patients all the way through to live birth. So this is quite challenging for many studies to make this link there. Of course, we cannot be sure, and as I showed the study from Dr. Kahraman, they actually did show that mosaicism can be present in the live birth. But I think that the incidents of this are rare, and some studies like the study by Biopsy have actually followed up on this, and they did look at some samples, some live births, and also some products of conception in particular. And we are seeing really low levels of mosaicism in newborns. But of course, not all newborns are being screened, and many of these mosaic abnormalities would not show a distinct phenotype. So because we are not sequencing, we may be underestimating the rate of mosaicism with live birth and in general in the population. But I think that generally these results are not exceedingly high in these mosaic abnormalities.
And when you perform the genetic counseling to your patients, when you are going to transfer a mosaic embryo, do you recommend to do a prenatal test during the pregnancy or not?
Yes, normally it is recommended, and normally amniocentesis is recommended because this is really what is the closest to the fetus. So we recommend amniocentesis.
Okay.
So also with diagnosing mosaicism, the patients do have to undergo amniocentesis, and this of course also involves or has its own risks.
So in that sense, when you transfer a mosaic embryo, the patient have to sign an informed consent that maybe they will have a really true mosaic.
Yes, yes, pregnancy. They have to be counseled about this, of course, yes.
Okay, perfect. Mina, you have already said that there is a very low rate to find a mosaic in live birth. But in your study in 2018 in human reproduction, when you study all the embryos that were donated for research, you find a 13 or 15% of true mosaic in the inner mast cells. What do you think about that?
Yeah, so that's a really good question. We do see mosaicism present at the blastocyst stage. But again, I have to mention that it all depends on the way the samples are analyzed. So when we looked at mosaicism, we also diagnosed mosaicism within the samples. So this can also indicate that the level of mosaicism within the inner cell mass is not so high. And there have been some recent studies suggesting that maybe embryos have some type of self-correction mechanisms. Now, we still don't have direct evidence for this, but we can, from cell lines and from cells in general, we know that cells that are abnormal have slower proliferation rates. So it could mean that there is some kind of out-competition of the euploid cells throughout development. And this could be why we are seeing lower rates of mosaicism throughout as development progresses. Of course, again, in our study as well, and I will mention it once more, we can't always exclude all technical variability. So in some cases, of course, when we show true biological mosaicism, then we can maybe assume that there is some self-correction, but in other cases, it could also be technical.
Ok. ¿Alguien tiene alguna pregunta para hacerle a Mina?
Yo tengo una, sí. Mina, regarding the origin of mosaicism, what laboratory practice could be related to an increase in mosaic embryo rate, since differences have been seen between centers, you know?
Yeah, there have been differences, and it has been suggested that certain laboratory practices can be associated with mosaicism. This is very difficult to answer because these studies, I mean, there are so many variables, so it's very difficult to establish one single variable and to exclude all the confounder effects. So studies like this are exceptionally difficult, and at the moment, I really can't give you a good answer. I mean, there are many things that we do that we know impact the culture of embryos in the lab. And it has been suggested that that it could be more related to the way we perform the biopsy, the use of the laser, for instance. So this is also... Sorry?
The quality of the cells.
Yeah, exactly. The quality of the cells, the quality of the embryo. So this all can have, of course, effects, and it can have effects both on just generating these artifacts, but also the culture, of course, the culture that we do, the pre-implantation culture. There could be many effects that may lead to these mitotic errors occurring. So suboptimal culture conditions or changes in temperature or pH. I mean, there are so many variables that we could look at. And I think it would be very difficult to design a study to really pinpoint any of these.
Yeah. And the second one, I really want to know your opinion about it. But what do you think about the detection of mosaicism and the non-invasive Pgta?
This is a very good question and a very difficult one too. So with non-invasive Pgta, what we see so far, I mean, detection, we're not detecting mosaicism with non-invasive Pgta. I think we're not really there yet. Because now non-invasive Pgta is really just being used to diagnose euploid and aneuploid embryos. And actually, it's been suggested as a better approach because this technique is not still at the levels of accuracy that we would like to see and not at the level of accuracy as a transferencia biopsy. We're really using it more as a system to rank embryos. So with non-invasive Pgta, what we would like to do is actually to keep all the embryos still in the running, where we just rank them. So it's another way to just maybe get to a better diagnosis on which embryo to transfer first. So at the moment, I haven't really seen studies looking at mosaic diagnosis from non-invasive Pgta. I think this is quite difficult, again, due to all the technical issues. I mean, with the transferencia biopsy, we're already limited because we have a limited amount of DNA, but at least this DNA is from cells and obtained directly from cells. Whereas the non-invasive Pgta, we're analyzing the media. So there is a lot of issues there. And as I mentioned, we're still not quite there yet, even for diagnosing just a binary euploidy and aneuploidy. So I think that diagnosing mosaicism would be exceptionally difficult. And I would like to see actually some studies that are really looking at this and correlating the media to different parts of the embryo. I think that this would be very interesting.
Mina, you have already said also that artifacts could be generated during the biopsy. So have you already read about if there is a type of mosaic embryos related to a bad biopsy? I mean, there is a correlation between a very poor biopsy and any type of mosaic embryos?
Yeah, so there have been some studies done where they were comparing like levels of noise in the sample to different biopsies and so on. But what has really been concluded is that if the biopsy is performed by an experienced embryologist, we actually don't see that much differences, and the level of noise is quite, consistent, or is quite okay. Of course, there are things that could impact this, as I mentioned already, the quality of the cells, the quality of the embryo. I mean, there are things that could impact, of course, maybe the time taken to tube the samples and so on, which can lead to DNA degradation. And of course, this could lead to some technical artifacts. So it's just something to be mindful of. Now, what specific types, and is there, I think with more and more samples that we analyze that have a mosaic diagnosis, it would be very interesting to see if there is a specific bias that we can identify. If there is a specific bias for a certain chromosome or a type of abnormality, this would be very interesting. But I think in the case of the segmental abnormalities, I think here, for me, at least in my opinion, there is a bit of a question as to what extent this is really mosaicism and what extent is it technical. And I think this is also a very difficult question. I mean, I am not arguing that mosaicism is biologically not present. I do think it is to a certain extent, but in some ways, I think we may be overestimating it with these technical limitations of the technique. So I think it would be very good to see studies that are looking at really this biological variability, maybe using other techniques or techniques other than the NGS.
Perfect. ¿Alguien tiene alguna pregunta más? Maxi?
No, yo no tengo más preguntas. No sé si alguien quiere hacer algún comentario o
Creo que hay una pregunta más en el chat, Maxi, me parece.
A ver. Mina, here, Mariana is asking if there is a relationship between that kind of the time of culture, if there is related to a higher rate of mosaicism.
No, we haven't actually seen this. So the time of, I mean, I assume you mean the time of biopsy. So like day 5, 6, or 7, we haven't seen any differences in rates of mosaicism related to the time of biopsy, or that is the time of culture. So at this stage, no. But again, yeah, these results are based on different, I mean, on very low, there's low data. So we are still unsure.
Okay. And I believe this is natural, maybe. The next question is related to the type of biopsy. If there is a differences between the flicking or the normal biopsy and the type of mosaic, or maybe the power of the time of pulse to do the biopsy, if there is a relationship between this and the mosaic embryos.
Yeah, there were some small studies reporting that the use of the laser could have an effect. But so far, what we have seen from the studies that are published is we are seeing no effect. Now, one thing to be mindful of course, is that the studies that are being published are published by labs and centers that perform Pgta a lot. So they're quite experienced with it. And this is something that we should, I think internally, it would be very good to look at rates of mosaicism. And perhaps it can even to some extent be used as some kind of performance indicator to see if there are some differences. I think in general, as I said, a well-trained and experienced embryologist, there shouldn't be any differences in the types of techniques. But again, we don't have enough data to really be able to make any definitive assumptions here. Yeah.
Mina, when we start doing NGS 2015, and we start to recognize mosaicism, I remember, you know, we detect a mosaic, we are so afraid to transfer. No, in that moment, we don't know nothing about mosaic embryos. Just the Monet studies when he did fish, and you know, it was so different between this and NGS. Do you think 6 years later, 2021, we're in a different point of view related to the mosaic embryo transfer, or we think that we have to continue, go step by step in this point?
Well, I think we're doing better in some ways, because I mean, I do agree with you. And I also remember looking at profiles and, you know, everything. I mean, the profiles that we often see also in publications are not, I mean, are much nicer than what we sometimes look at. So, I mean, it wasn't easy, but I think that in some ways, some providers and some laboratories that are really doing a lot of sequencing have to an extent improved their algorithms. There have been algorithms that are now being designed specifically for diagnosing mosaicism. As I mentioned in the beginning, the algorithms were all really made to just diagnose aneuploid or euploid embryos. So we're really relying on subjective observation without really much knowledge or, to make educated decisions. So I think now the more we are exposed to these kinds of diagnoses and the more data that is being released on this, we are getting a bit better. And as I said, we are improving our algorithms. There are algorithms that are specifically made for calling mosaic diagnosis. So I think in these ways we are getting better. But I think in terms of, knowing, as I said, the extent to which it's biological or technical, and to knowing really the developmental potential of all of these mosaic abnormalities that we are diagnosing, I think this is still, we are not there yet. I mean, I think there will still need to be a lot more clinical data and, as I said, more prospective studies, which are really the least biased, that will give us some more insights on this.
And finally, in that sense, do you think that artificial intelligence will help in this field too?
I think it may help, but again, we are limited with what we are observing. So I think, to an extent, potentially, I mean, it has some merit, but again, as I said, we are limited with the technology itself. So artificial intelligence cannot overcome that. And it is only based on kind of mostly these static observations. So like we see with the, morphology. So I'm not sure. I think there is some, I mean, it's definitely an area to explore. But I'm still, I guess, still skeptical.
Perfect. Thank you so much. Bueno, Maxi.
Sí, estamos, creo, ya ¿no? Para cerrar, ¿no?
Buenísimo.
Si no hay más preguntas, cerraríamos ahora directamente.
Well, Mina, thank you so much for your time. We really appreciate this. We are very excited about your data, as I told you before. Thank you so much for your time, I think it's late right now in Barcelona, but thanks again. And maybe we will want to share with you this data in the future, maybe in the SAEC Congress.
For our Congress, yeah.
Yes.
For our meeting in November, yeah.
In November.
Sure, it would definitely be a pleasure. And thank you very much once again for inviting me. And I hope to see you all soon.
You're
Perfect. Thank you so much. Gracias a todos por su presencia. Y bueno, los esperamos en nuestro próximo webinar. Gracias, Maxi.
Gracias a vos, Cristian. Nos vemos. Hasta luego.
Gracias a todos.
Gracias a todos.
Gracias.
Thanks, Mina.
Thank you.